If you look at the learning objectives, obviously we're not going to get through the entire chapter. We're again covering like the first third of the chapter and that's it. So we'll be talking about portals of entry and exit, and then we'll talk about once a microorganism is into the body or inside the body, what happens next, as to how the microorganism starts to mount an immune response, or how the immune system starts to mount that immune response. Okay, portals of entry. Again, this is kind of a review at this point. Portals of entry, this just simply means how microorganisms enter into the body. So one of the main ways that they enter into the body is through mucous membranes. So you have conjunctiva, that is the eyes. So we talked about several parasites that can enter directly into the eyes. Certain viruses, certain bacteria can enter straight into the eyes. That's why we have pink eye, and we have styes. Those are due to some type of microorganism. Then the main portal of entry next is respiratory. And you see it has the little asterisk next to respiratory and the GI tract. These are the two main portals of entry. These are known as the preferred portals of entry, because although a microorganism may be able to enter the body through several different portals of entry, generally the respiratory tract or the GI tract are considered the preferred portals of entry. So respiratory, this is through droplet inhalation of moisture or dust particles that contain these microorganisms. It is the most common portal of entry. Make sure you know that for the exam. So the respiratory tract is the most common portal of entry. This is in the news a lot right now, simply again because of the coronavirus. You contract the coronavirus, you contract the regular flu, you contract the common cold. There are quite a few diseases that you contract through inhalation. And so we'll be talking about more as to what happens once the microorganisms gets into your respiratory system. Another common portal of entry, though, is through your GI tract, and this is through contaminated food or water. So we've talked about food that's left out. We're always hearing about food being recalled due to some type of pathogen such as E. coli or Salmonella or Shigella. We talked about parasites living in water. So hikers that go backpacking, they drink water that they think is clean and it turns out not to be clean. Or simply through contaminated fingers. We don't wash our hands like we're supposed to, and the coronavirus is proof of that, as far as spreading. So contaminated fingers, there's all kinds of microorganisms that are on our fingers, and we tend to put our fingers in our mouth more than we realize that we do. And so this is another very common portal of entry. Next is genitourinary. Okay. Now, genitourinary is more common in females than in males. However, it can happen in males as well. So if you are familiar with UTIs, urinary tract infections, okay, that is considered genitourinary. Women sometimes will come down with an illness known as bacterial vaginosis. Again, that is considered genitourinary. Now, there's a mucous membrane, or there's a portal of entry that we know that we've talked about that's not even on this list, and that is the nose. Okay. So we talked about respiratory tract through inhalation, but remember, we also talked about a unique parasite, Naegleria fowleri, that enters into the nose and it travels to the brain. Okay. So that is another potential portal of entry. Okay, moving on and talking about skin. Skin is the largest organ of the body. Our skin is very good at keeping microorganisms out. That's the job of the skin, is for protection of our internal organs. But it is possible for potential pathogens to penetrate the skin through hair follicles and sweat ducts. And then there are some pathogens that infect the skin itself. We've talked about ringworm. We talked about athlete's foot. There's another common disease called impetigo that affects the skin. So that is also another portal of entry. Lastly is parenteral. This may be a new term for you. Parenteral means that there is a break in the skin. Okay, this could be caused by trauma. So this could be where you scrape your knee, you step on a rusty nail, and now you have an opening. It's bleeding. That is now considered an open wound. That is a parenteral root. So you have that scratch that is now open to the environment. That is considered parenteral. Okay, we also know that arthropods bite, they sting. We talked about one of the main arthropods being mosquitoes, and how they bite and they deposit the potential pathogen into the host. We talked about mosquitoes when we talked about yellow fever, when we talked about the Zika virus, when we talked about heartworms, when we talked about the plasmodium, which causes malaria. So that is also considered a parenteral root by the use of arthropods. And then lastly is injections. So this is with some type of needle. So if you ever go and have blood drawn, they will swab the area with an alcohol wipe first. The purpose here is to kill any pathogens or kill any bacteria that may be around the site where they are about to puncture the skin. So injections, anything that breaks the skin is considered parenteral. Okay, well, if we have portals of entry, we have to have portals of exit as well. And these are basically the same as the portals of entry. So if these microorganisms enter the body, well, they're going to have to leave the body as well. And a lot of times they leave the same way in which they came. So one of the main portals of exit, once again, is the respiratory tract. I said last lecture, a typical a typical sneeze contains 10,000 droplets. A typical sneeze can spread about 6 feet, although they are now saying that with a very forceful sneeze, it can spread up to 24 feet. So that is definitely a portal of exit. And you can see some of the diseases that are spread via the respiratory tract. We have tuberculosis, which we've talked about, whooping cough, pneumonia, scarlet fever, meningitis, chickenpox, measles, mumps, smallpox, flu, and now we can add the coronavirus to this. So there's a lot of these that we have already talked about this semester. Moving on, the gastrointestinal tract. This is another biggie here. This is another main portal of exit. This is usually through fecal oral. This is where somebody has went to the bathroom or there has been raw feces, raw fecal matter that has somehow gotten into the environment. And then they use that same drinking water or that same water as drinking water. So in countries that don't have proper sanitation, gastrointestinal diseases are very, very common. So a couple, salmonella, cholera. We talked about cholera the last class with the last lecture I gave. Typhoid fever. Remember Typhoid Mary, the story of Typhoid Mary. Shigellosis, amoebic dysentery. We've talked about dysentery, polio, rabies, mumps, infectious mononucleosis. So again, this semester we've talked about most of those. Genitourinary tract, urine and vaginal secretions. Okay, once again, with urine, it could be a urinary tract infection. With vaginal secretions, it could be bacterial vaginosis, or it could be a sexually transmitted infection such as chlamydia or gonorrhea. So those are very common vaginal pathogens that can be found in vaginal secretions. Lastly, or next, is skin. Skin can also be a portal of exit. We talked about how ringworm is usually on the extremities, arms or legs, and it's highly contagious and it lives on the skin, and you scratch it and you wind up with the fungal cells under your nails, and then you pass them on to a neighboring person. Maybe you shake somebody's hand and you pass that disease on to them. So that is how skin can be seen as a portal of exit. And then lastly, again, with the blood, the biting arthropods and needles or syringes. With biting arthropods, such as a mosquito, they don't just bite one host, they bite multiple hosts. So it's possible that they picked up one parasite in one host, they carried it to a new host and deposited the parasite. Or as with the use of needles and syringes right now. Over the past few years, we've been facing an opioid epidemic where certain opioids, their use is on the rise. And there are a lot of communities that are suffering from very high rates of certain diseases like hepatitis B, hepatitis C, and HIV because they're reusing needles. So reusing needles would be considered a portal of exit. So there are three variables that we are going to talk about. The first one being what is known as R sub 0, but it's traditionally called R naught. And then on the next slide, we'll move in and we'll talk about ID50 and LD50. So R naught, it stands for the basic reproduction number. It talks about how contagious a particular pathogen is. So the example is if R sub 0 or if R naught is 2, then one infected person is expected to infect 2 other people on average that are susceptible to the disease. So that means the other population of people, they cannot be vaccinated or they cannot be immune to the particular pathogen. So if 100 people were contagious, it talks about how many people, if one person has a disease, how many people could that person pass it on to? So typically, if an R naught value is less than 0, it means that that disease is not particularly contagious. And at some point over time, it will kind of die out. Now, if the R naught value is closer to 1, it means that the disease may remain stable in a population for some period of time. It may not go away right away. And then what's the scary one is if R sub 0 or R naught is greater than 1, it means the disease is likely to spread and could become an epidemic. This is exactly what happened with COVID. They gave COVID an R naught value of somewhere between, I think it was 2.5 and 3.5. So COVID had a really high R naught value. So basically what that means is for every person, they would pass it to potentially 2 to 3 other people. And that's why COVID became an epidemic so quickly. Okay, so there are 2 terms that you need to know. Make sure you know both of these terms for the upcoming final exam, the ID50 and the LD50. The ID50 it stands for infectious dose 50, meaning it is the infectious dose for 50% of the test population. So let me give you a particular example. If I have 100 people in a room, if I were to release the coronavirus into this room that had 100 people in there, how much of the coronavirus would I have to release into the room for 50% of those people to get sick from the coronavirus? So again, if there's 100 people in there, I would need 50% or 50 of those to become sick. So how much of the disease has to be present for 50% of the test population to become infected with that particular disease? Okay. So ID50 levels are very, very crucial when it comes to the different diseases out there. We need to know how much disease has to be present before 50% of the population is going to come down with that particular disease. Just as important, maybe even more important, is lethal dose 50. And remember, this mainly deals with toxins because a person oftentimes, they don't really get sick from the bacteria itself. They get sick from the toxins that are produced by the bacteria or by the virus. So with lethal dose, you're talking about the lethal dose of a population for 50% of the population. What that means, lethal means deadly. So you're looking at the deadly dose. LD50. How much of a toxin has to be present before half of the test population dies? Well, we obviously cannot use LD50 levels on humans because it would require that half of the test population died. However, we have done research using mice and rats and other animals trying to figure out the LD50 level. Every new medicine that is trying to be passed by the FDA to go onto market, you have to know the LD50 levels of that medicine. The LD50 levels gives you information on overdose levels. An overdose, typically an overdose, they get overdose levels or overdose information by using the LD50 level. What percentage of a drug would kill has to be given before half of the population would die from it. So most of the medicines that are currently on the market that are prescription based, they've had LD50 studies performed to see overdose. How much of that drug would a person have to consume before half of the test population would die? So both of those terms, the ID50 and the LD50, both of these are very, very important. Now, if you look at Bacillus anthracis, this is the bacteria that causes anthrax. We know that it is a very deadly bacteria. There's been a lot of research done on it. It's been used as a method of bioterrorism in the past. Robert Koch, he did most of his work with Bacillus anthracis. Now, the interesting thing about Bacillus anthracis is we all know, I've already said it's deadly. Bacillus anthracis is very deadly. But depending on the portal of entry depends on the infectious dose. So let me give you the example. If you look at this chart here, the portal of entry, if I were to take a knife and cut a line in my skin, if I were to deposit Bacillus anthracis directly into the open wound, the ID50 level is 10 to 50 endospores, meaning that it would take about 50 endospores for half of the population to come down or to become infected with Bacillus anthracis. However, you can take that same Bacillus anthracis, and if you were to inhale it, well, now it takes up to 20,000 endospores to reach that same level of ID50. And then lastly, if you were to ingest Bacillus anthracis, it would take up to 1 million endospores to cause that same ID50 level. So remember, the ID50 doesn't change. This is the infectious dose for 50% of the population. This shows, though, that depending on the portal of entry used, it may need less numbers, such as with the skin, it only needs about 50 endospores to cause ID50. But if you were to eat Bacillus anthracis, it could take up to about 1 million spores to cause the same level of infection as 50 endospores directly deposited into an open wound. Okay, toxin examples. So over here on the left, we have botulinum. It's a toxin, a very toxic toxin. Just a note about botulinum. It is the most toxic, naturally occurring substance on the face of this planet. That is a test question. Again, the botulinum toxin is the most toxic, naturally occurring substance on the face of this planet. Okay, so botulinum. This comes from the bacteria Clostridium botulinum. Okay. We've talked about Clostridium in the past. This is one of our endospore formers. And botulinum, the botulinum toxin, this is the same thing that they make Botox out of. Botox is literally botulinum toxin. So I just said that the botulinum toxin is the most toxic naturally occurring substance, and that's what they make Botox out of. So if we go below the botulinum toxin, you see Shiga toxin. We haven't really talked about Shiga. The Shiga toxin comes from a bacteria known as Shigella. This is one of the main 8 causes of food poisoning here in the United States. So these Shigella bacteria, they produce something known as the Shiga toxin, and when consumed, it's not so much that you get sick from the Shigella bacteria, it's that you're getting sick from the Shiga toxin that the Shigella produces. Lastly is the staphylococcal enterotoxin. Well, we've talked about staph quite a bit. There's multiple strains of staph, there's multiple types of staph, but most of these staph, they produce toxins. And it is the toxins that cause all of the nasty side effects. But if you look over at the right, you see the ID50 for these 3 different toxins. For botulinum, you only need 0.03 nanograms per kilogram to reach an ID50. But if you were to look at the Shiga toxin, you require 250 nanograms per kilogram to reach that same ID50. And then lastly, if you look at the staphylococcal enterotoxin, you need 1,350 nanograms per kilogram to reach that same level of ID50. So which is the least potent toxin? Well, the least potent toxin is the staphylococcal enterotoxin, simply because you need the most of it to reach ID50. Okay, so once the pathogen has entered into the body, now what happens? Well, it turns out that most of these pathogens have something known as adhesins. There's 2 types of adhesins. First of all, these adhesins for the most part are protein-based, but there's 2 types. You have your glycoproteins. There are remember the prefix gly means sugar. So these are sugar-based proteins. And then you have your lipoproteins. These are your lipids or your fat-based proteins. So the pathogen has these adhesins on its surface. Okay, so if you look at this picture here, let me turn on my marker. If you were to look at this picture, these are the adhesins, the things I'm circling in red. Okay. So this looks like a typical bacteria cell, and then attached to the bacteria cell, you have these adhesins, which are protein-based. Along with adhesins, if you go way back to when we were talking about cell anatomy, a couple of other factors that play a part in the adhesins, because it says down here, adhesins can be a part of, remember we talked about the glycocalyx, that the glycocalyx was a sugary sticky substance secreted by some cells, and it aimed in attaching to other host cells to make up a biofilm. And we also talked about fimbriae, which were long, thin hair-like projections that worked along with the glycocalyx to form those biofilms. So you find these adhesins, you can find them as part of the glycocalyx, you can find them as part of the fimbriae on the pathogen. And when the pathogen enters the body, the adhesins on the pathogen are going to bind to host cell receptors. So we could be talking about us. We could be the host. Okay? So pretty much on every cell that we have unique receptors. Some of those, first of all, we have a variety of receptors. But a certain type of receptors, there's just a simple sugar receptor known as mannose. So again, mannose is a sugar. And we have receptors on our cells. Most host cells have receptors made out of mannose. And what happens is when a pathogen enters into the body, the adhesins on the pathogen bind to the mannose receptors on the host cell. So that is how the initial binding occurs between the 2. Okay. And again, we've already talked about biofilms and what they do. We've talked about them in a couple of different chapters now. So on the previous slide, we talked about adherence. Once a pathogen enters the body, the first thing that it has to do is adhere. After it adheres, then it has to find its way to make its way into the cell or near the cell of target to cause infection. So a pathogen must be able to invade the appropriate tissue and obtain nutrients. Once a pathogen enters the host and adheres to tissue, it must obtain nutrients, like I said. There is a type of organelle, basically. It's somewhat similar to an organelle known as a siderophore. These are known as iron stealers. One big component is iron. Most bacteria need iron to survive. Well, we as humans, we do not have a lot of free-floating iron within our body. Our iron is normally bound. So these bacteria, they contain a component known as siderophores, which basically steal the iron that is bound to our body. So that's one way that they are able to obtain the iron that they need. So following adhesion, the pathogen has several options. It can stay on the surface of the host cell and damage the surface layers of the host cell, eventually causing death due to damage of the plasma membrane or the cell wall. However, some pathogens have the ability to actually pass through the cell wall, get inside to the center of the cells, and invade deeper tissue and cause cell damage on the inside. Some pathogens are able to enter the cell and they reside inside the cell, like we talked about with some of the viruses and how they're able to live inside cells. They live inside cells and they produce toxins and they react from inside of a host cell. As pathogens invade, they tend to damage host tissues and generate cytopathic effects. So cytopathic just means cell changing. These are bad toxic byproducts or cellular component products where they're able to change, such as the cell wall or the plasma membrane. So here on the next slide, like I was saying, here on the next slide, this shows how a pathogen must invade the tissue to obtain nutrients. So some of these pathogens, like at the top of this picture, you can see they kind of stay on the receptors of the host cell. Okay. Whereas others are actually able to move inside the cell and they cause problems from inside their cell. They're intracellular. We talked about a couple of the parasites that were considered to be intracellular. The group Apicomplexa, which was group number 4 that I talked about, which included the Plasmodium parasite, as well as Cryptosporidium parvum. We talked about toxoplasmosis and Babesia. Those were all considered to be intracellular parasites. So they live inside of a host cell and they're still able to cause damage. Well, that's the same with some other pathogens, such as the bacteria and the viruses. They're able to invade a host cell and they cause damage from being inside the host cell. And then lastly, you can see at the very bottom, there are some pathogens that actually pass all the way through the cell layers to get to the inner tissue, and they can invade tissue and muscle, release toxins, and cause a problem from that aspect. Okay, so once the pathogen has entered into the host we have immune systems, and our immune systems are designed to kill the invading pathogen. However, some of these pathogens have gotten really smart over the years, and they have developed mechanisms that prevent them from being exposed or prevent them from coming in contact with the host immune system. So we've already talked about capsules quite a bit. Remember, the purpose of a capsule is that it prevents phagocytosis. So the capsule is the sugary sticky substance, the glycocalyx. There are certain bacteria that have a capsule around them, and if that capsule is present, phagocytosis cannot occur. So remember, the host immune system has cells known as phagocytes. These are cell eaters. And the phagocytes are released and they circulate in the blood and through the lymph, and they attempt to come upon invading pathogens. They'll surround them and kind of bring them in and eat them or break them down and degrade them. But if that invading pathogen, if that is a bacteria cell that has a capsule around it, phagocytosis can't occur. So it will allow for that particular bacteria to continue to survive. Another thing is cell wall proteins. This is a very specific example is using the example of S. pyogenes. This is Streptococcus pyogenes, once again, the bacteria that causes strep throat. Well, Streptococcus pyogenes has something known as M protein. And M protein is a very large protein. It's an unusually large protein. The purpose of M protein, when it's present, is once again, it's so large that phagocytosis can occur. So these phagocytes are floating around the blood and the lymph, and if they were to come upon Streptococcus pyogenes, they would attempt to phagocytize the S. pyogenes. However, S. pyogenes has M protein, this really large protein. And so phagocytosis, that phagocyte is small enough that it can't wrap around the M protein and engulf it and break it down. So once again, it's a means or mechanism of survival. Now, another mechanism of overcoming the host defense is through antigenic variation. And antigenic variation, okay, I'll go ahead and say, I think antigenic variation is kind of cool. It's very, very neat the way these bacteria or these pathogens are able to perform antigenic variation. Now, again, not all microorganisms can perform antigenic variation, okay? But antigenic variation is the avoidance of the IS, which is the immune system. So antigenic variation is a very smart way of avoiding the immune system. I gave two examples on here, Trypanosoma and Neisseria. Both of these genera are able to perform antigenic variation. We've talked about Trypanosoma when we talked about Trypanosoma brucei and Trypanosoma cruzi. And then we talked a little bit, I told you guys about supergonorrhea, the sexually transmitted disease. It is caused by Neisseria. So with antigenic variation, what happens is the host immune system elicits an immune response that is very, very specific. So I'm going to label this one here A. So Trypanosoma or Neisseria enters into the host, and we're going to use a human as a host here. Well, the immune system of the human is going to recognize A, and it's going to mount an immune response to A, and only A, because I said these are very specific. The host immune response is very, very specific. So our immune system, the person's immune system, will mount a response towards A. But before that response can be completely elicited out into the environment, look at the surface projections of the pathogen. Well, it changes shape. It goes from these rectangular-like structures to triangles, and we will call this B. Okay, so the immune system has mounted a response towards A and only A. So what does A do? A changes to B. Now our immune system won't do anything because our immune system is targeted to A, not B. So our immune system has to go back to step one and reconfigure and reorganize an immune response towards B. And while that's taken place, the shape of the receptor will change again. It will go from a triangle to a half circle or an arc. And we'll call this C. So our immune system at this point has mounted a response to B and only B, but B is changing to C. So our immune system can't do anything about C because it only mounted an immune response towards B. So now it has to go back a step and mount an immune response towards C. So with antigenic variation, it always stays one step ahead of the immune system. Okay. It is estimated that with some of these, when antigenic variation occurs, that there's over 1,000 different variations that those surface receptors can change to. So that is how they are always thought to stay one step ahead of the immune system, because the immune system will mount a response towards one thing, and that antigen is changing shape to something completely different. So now our immune system won't work. So that is what I think. That's why I was thinking it was kind of neat how antigenic variation occurs, because this allows for the survival of the pathogen. And then lastly is penetration into the host cell cytoskeleton. Okay, host cells, these are typically eukaryotic cells. Eukaryotic cells have very intense and very strong cytoskeleton networks. This is the cytoskeleton is what helps maintain the shape, the function, and the structure of the eukaryotic cell. So there are some bacteria, such as Salmonella and E. coli. These are both foodborne bacteria, very common causes of foodborne illness. They produce something known as invasins. Invasins, these are proteins, but they're toxins at the same time. These invasins help move the cytoskeleton. One of the main components of the cytoskeleton is a protein known as actin. Okay. And normally the cytoskeleton of the cytoskeleton of the host cell, the eukaryotic cell, is usually a very dense structure. It is not easy for molecules to enter from the outside of the eukaryotic cell to the inside of the eukaryotic cell due to the cytoskeleton. However, if E. coli or Salmonella were to attempt to get inside of a host cell, it would release invasins. And basically, once again, it allows movement of the cytoskeleton so that now it's easier for these bacteria to penetrate inside the host cell. And I think that's seen on the next slide right here. Here's a picture of invasins. Okay, again, Salmonella and E. coli. They produce those invasins. Those invasins cause what's known as ruffling. It's basically where the cytoskeleton, the configuration of it changes to now allow movement of the cytoskeleton, so it moves out of the way so that the bacteria can enter into the host cell without damaging the host cell. And believe it or not, that is where I'm stopping with chapter 15. So again, this is the first 9 slides of chapter 15. I don't even usually cover this here at the bottom. I don't even cover that. So again, this is all that you have to know for chapter 15. The next lecture that I will send out will be the first half of chapter 20. And again, that will probably be Tuesday. All right. You guys make sure to let me know if you have any questions. So I'm going to stop here for tonight. Y'all have a good night and happy studying. So let's look at some of those invasins in a little more detail. So the first thing that you should notice about the invasins is that they are enzymes. And we know this because if you look at the names of the invasins, they end in the letters ASE, which means that they are an enzyme. So these are different enzymes that allow pathogens to invade host cells. The first one being coagulase. So it helps with blood clot formation. It protects bacteria from undergoing phagocytosis. So we've talked about phagocytes a couple of times throughout the semester. Phagocytes, these are known as cell eaters. And they're a very important part of the immune system. They're always available, but there are certain bacteria and certain pathogens that release coagulase. And again, this prevents our immune system, our phagocytes, from being able to eat the potential pathogen. Kinase works to dissolve blood clots. An example, streptokinase. This becomes a problem because sometimes the pathogen actually gets caught in the blood clot as the blood clot forms. So when kinase is released by the bacteria, it dissolves that blood clot, allowing that bacteria to now roam free. It's no longer caught in that blood clot. The third one being hyaluronidase. Yes, it's a mouthful. It breaks down hyaluronic acid, which acts as glue to hold connective tissue together. All right, so it's an enzyme and it's released by certain pathogens, and it basically breaks down connective tissue, allowing pathogens to enter into areas that at one point they couldn't enter into due to the connections that were held together by the hyaluronic acid. Um, Collagenase, has to do with the breakdown of collagen hydrolysis. So basically, it's collagen with the formation of water. It facilitates the spread of Clostridium tetani, which causes gas gangrene. And then you have the neuramidases. Yes, a lot of these are really long words, but it breaks down neuraminic acid, which is important in regulating host cell communications and membrane transports. Going on to the next slide, you can see some of those enzymes in action. So at the top you have the coagulase. It's basically causing it to coagulate, trapping the pathogen. But then with the streptokinase, it dissolves the blood clot, allowing release of the pathogen. And then at the bottom, you see hyaluronidase and how it breaks down that connective tissue barrier, allowing those bacteria to seep in through the middle because they're no longer connected together because that they were broken down due to that hyaluronidase. So now the pathogen is inside the host cell. How does the pathogen actually damage the host cell? And again, we've kind of talked a little bit about this. I said it uses host nutrients, such as our iron supplies. So those are those siderophores that I were talking about. They are iron stealers. So they basically, in a way, they kind of act like viruses. They kind of take over. They take what they need for their own use. Okay. So anything that they need, they've evolved a way to pretty much take what they need for survival. Then some of these bacteria or some of these pathogens cause direct damage. They may break through the cell wall, or they may lyse the plasma membrane. They may dissolve the phospholipid bilayer. They start breaking down proteins, which ultimately leads to a weakened bacteria cell or a weakened host cell, and it allows that host cell to break apart and dissolve. Produces toxins. Okay, we have not talked about the toxins yet, but later in this chapter, in just a few minutes, we'll start talking about toxins. There's two main classes of toxins. You have your endotoxins and you have your exotoxins. And again, we'll move on and talk more about that. 4, induce hypersensitivity reactions. If you know anyone with asthma, asthma is considered a hypersensitivity reaction. It's where our immune system is overly stimulated, and it sends out a greatly increased reaction. Okay. So our immune system is really great. Our immune system encounters pathogens every single day, and for the most part, our immune system is really good at fighting off of these pathogens so that we don't get sick. However, over time, these pathogens have evolved ways in which to survive our immune system or trick our immune system into not recognizing the pathogen. So there's a couple of different approaches here. One way is that the pathogen can hide, okay? And if it's hidden, then our immune system can't find it. And so examples of this including things like antigen masking, mimicry and variation, latency, and intracellular lifestyle. Well, we've kind of already talked about intracellular lifestyles because we know from previous talks that there are a variety of different pathogens that can live inside the host cell, meaning the intracellular. So they cause the damage, but our immune system can't find them because they are inside one of our cells. Okay. Antigen masking is basically, this is where the antigen covers itself in host antigens to avoid immune detection. So basically the antigen looks like it belongs there. There is nothing for our immune system to detect because that particular antigen got really smart and or the pathogen got really smart and figured out how to cover itself with the antigen. Another technique is through mimicry. And this is where the pathogen, again, the pathogen is smart. The pathogen makes the antigen. It manufactures the antigen that resembles the host antigen. And therefore, again, the immune system doesn't see it as foreign because it looks like all the other cells. So those are some ways in which the immune system is not so good because these pathogens over time have evolved mechanisms to survive. So sometimes if a pathogen can't avoid immune detection, it does have the ability to limit the immune system's actions. And basically, there's a couple ways of going about this. One is by suppressing the immune function. So basically, it makes your immune system not work at 100%. It suppresses your immune system. Another way it can do this is by, again, avoiding phagocytosis. So those phagocytes, those cell eaters, are present, but the pathogen has evolved ways so that the phagocyte is not able to consume it. It's not able to eat that cell like it's supposed to be able to do. And here, this picture shows you different ways in which the pathogen has the ability to escape phagocytosis. It's evolved several different ways of doing so. We are about to begin our talk on toxins, those exotoxins versus those endotoxins that I mentioned a few minutes ago. However, before we start talking about toxins, let's just talk about a few terms that are going to be related to terms that you hear in the upcoming few minutes. So a toxin, this is simply a poisonous substance that contributes to pathogenicity. Okay. Toxogenicity is simply the ability to produce a toxin. Toxemia. We learned this from a previous chapter. Emia means in the blood. So toxemia means toxins in the blood. Toxoid. This is an inactivated toxin. So this is a toxin that does not function anymore. An antitoxin, these are produced in response to an exotoxin or its toxoid. Antibody produced in response to an exotoxin or its toxoid. So this, again, is part of the immune system. Here are those two main classes of toxins that we talked about, the exotoxins versus the endotoxins. The exotoxins are typically made out of protein. These proteins are usually enzymes. The exotoxins are found in both gram-positive cells as well as gram-negative cells. And these happen to be very soluble in body fluids, which is why they're able to become systemic very quickly, is because of their solubility. Also, there is one study that says they have discovered over 200 different exotoxins that could potentially harm humans. Now, on the flip side, the endotoxins are only found on gram-negative bacteria. And the endotoxin is called LPS, lipopolysaccharide. It's part of the outer plasma membrane. Remember, with gram-negative bacteria, you typically have three layers. You have your outer plasma membrane, you have a very thin layer of peptidoglycan, and then you have your inner plasma membrane. The endotoxins, they're found only on the gram-negative bacteria because only gram-negative bacteria have an outer plasma membrane, which is that phospholipid bilayer that we talked about. And actually, if you look at this picture here, you can see the phospholipid bilayer. Okay. So again, these are the main two classes as far as the endotoxins. Usually when a cell dies, or sometimes when a cell undergoes binary fission, endotoxins are released. These can be very, very deadly and cause death. because they can cause something known as endotoxic shock, which causes things like fever, chills. It can go into cause severe things such as convulsions, um, rapid heart rate, coma, and even death. So this is kind of a review of what we just went over. With the exotoxins, it affects both gram-positive and gram-negative bacteria. It's considered to be a byproduct of the cell as it grows. Most of these exotoxins are proteins. Like I said, there's over 200 different exotoxins that they have found, and the exotoxins are usually named after the system that they infect. So a neurotoxin infects the nervous system. Enterotoxins typically infect the GI system, and so on and so forth. They typically do not cause a fever. They can be neutralized by an antitoxin, meaning we have antitoxins available, which is very good because in a minute you'll see a list of potential exotoxins. And so we do have uh, cures or antitoxins available. So if a person were to come down with a sickness due to one of the exotoxins, we can neutralize the exotoxin with an antitoxin. The LD50 rate, remember lethal death 50, is typically small. So there's usually not a lot of people that die from the exotoxin, meaning it takes it's not over super deadly. They circulate to the site of activity, and they have the ability to affect the body before the immune system responds. And that is another reason why these exotoxins tend to be so severe, is because they're able to act very quickly before our immune system can act. All right, so here are the three main types of exotoxins. Okay, they're simply called type 1, type 2, and type 3. So at the very top, you see the picture of the bacteria cell. It's releasing the exotoxins. You see the host cell, you see the nucleus of the host cell. You see that those exotoxins have now come in contact with the host cell. Well, one of three things typically happens. With type 1, the toxin binds to receptors that are on the surface of the host cell. And then it creates a cascade effect, or kind of like a domino effect. The receptor is activated and then causes changes inside the cell. And a lot of times those changes are detrimental and can potentially cause cell death. Now with type 2, the toxin actually causes disruption of the plasma membrane. It causes the phospholipid heads to be chopped off of the tails, or it causes the plasma membrane to dissolve. So it affects the actual plasma membrane by causing harm to the plasma membrane. Type 3, okay, these are also known as the AB type, because the toxin itself has an A portion and a B portion, but they are linked together. And the binding portion is brought in through the inside of the cell through endocytosis, and then the active portion is the portion that exerts the toxigenic or the pathogenic effect on the cell. So those are the three main types of exotoxins. Again, type 1, which occurs with the toxin binding to receptors on the surface of the host, causing a cascade pathway to occur, which occurs on the inside of the cell, causing damage. There's type 2, which causes disruption or causes the breaking down of the lipids that form the plasma membrane. Or then there's type 3, which is known as the AB toxins. It's where the part B is actually what helps penetrate into the cell wall or that plasma membrane, but it's the part A that causes destruction of the host cell. So again, a summary of the main three types of exotoxins. Okay, so here's a list or representative example of some of the exotoxins. Several of these bacteria have been mentioned throughout the semester. C. diphtheriae, Dipsodiphtheria. This is one that we are still vaccinated for because in times past there were many, many people that died from diphtheria. Streptococcus pyogenes. I've mentioned this one. This is one of the main causes of strep throat. Well, it releases a toxin. Clostridium botulinum. We've talked about Clostridium botulinum because this causes botulism food poisoning, a very deadly kind of food poisoning. Clostridium tetani. This causes tetanus. So if you've ever stepped on a rusty nail and you had to go get a tetanus shot, it's because of Clostridium tetani. That is the fear here. Vibrio cholerae. This causes the disease cholera, which we talked about in the last chapter when we were talking about epidemiology and the outbreaks of cholera in the water supply. And then, of course, Staphylococcus aureus. It's an AB toxin. It's considered to be a super antigen, though, because it over or hyper stimulates our immune system. And that's why our immune system, that's why we get really high fevers. And that's why we have the signs and symptoms that we do. And why our signs and symptoms are heightened with Staph aureus is because it's said to be a super antigen. It not only stimulates our immune system, it overstimulates our immune system, causing our immune system to provide a really fast, really large reaction. And that's why really high fevers and other symptoms are associated with Staph aureus. Okay, so a brief review of endotoxins. I said there's really only one type of endotoxin as compared to the exotoxin. So there's 200 different types of exotoxins, but as far as endotoxins are concerned, there's really only one type, and it's known as LPS, which stands for lipopolysaccharides. These are the ones that are only found on gram-negative bacteria because the LPS is actually embedded in the outer plasma membrane, and only gram-negative bacteria have an outer plasma membrane. So you can see here in the picture, the endotoxins, these are basically released when the bacteria die. So at the very end, when the bacteria they lyse open, releasing that endotoxin. Also, as bacteria divide through binary fission, it is possible that a small amount of the endotoxin is released. These do typically cause a fever, but look, we do not have an antitoxin for it. Unlike the exotoxins, as far as the endotoxin, which is, like I said, the main one, LPS, we don't have an antitoxin for it. So this is where our immune system plays a large role. We have to depend on our immune system because we don't really have any type of vaccine or treatment for this. It has a relatively large LD50 level as well. Okay, so a little bit more. So on endotoxins. So endotoxins, these cause bacterial cell death, also antibiotics, because antibiotics cause bacterial cell death. So they target the bacteria die, they lyse open, and then the endotoxin is released, that LPS. The endotoxin is known to cause fever, severe fever, okay, chills, weakness, aches, and death. It mentions interleukin-1. We'll talk more about interleukin-1 when we get to the immune system in the next couple of chapters. If present in sufficient quantities, the endotoxin causes septic shock. And remember, we don't really have treatment for it. We don't have an antitoxin for it. It works as to basically causing organ destruction and causing the organ to fail. Septic shock kills 11 million people per year globally. This is considered one of the leading causes of death, is the endotoxin, that lipopolysaccharide that's found on the outside of those gram-negative bacteria. And you can see at the bottom, this is the process as to how the endotoxin causes a fever. Okay, so basically the endotoxin, which is on the bacteria is brought into the host cell. Okay, the bacteria or the host cell can't really kill it, can't really break it down, but it releases chemicals that go to the hypothalamus of the brain, which releases prostaglandins, and those prostaglandins, as they're released, they cause a fever. So that is the overall role in how these endotoxins are known to cause fever. It messes with the hypothalamus of the brain, which causes release of the prostaglandins. Okay. And the last slide of this chapter, you can see the microbial mechanisms of pathogenicity. It's kind of just an overview. We talked about portals of entry. We also talked about portals of exit because they're pretty much the same. We talked about once the organism or once the pathogen enters into the host, what happens? Well, it has to adhere. Okay, it has to adhere using adhesins. And then next, it will use invasins. We talked about the enzymes that are used as invasins to invade the host cell to cause disease. We talked about the differences between endotoxins and exotoxins and how they work to damage the host cell. Okay. And that is the end of this chapter.